The demonstration that we will present today is that of the sympathetically innervated cat atria. Now, the purpose of the experiments that we're going to do are threefold. Firstly, we'd like to demonstrate how a biological preparation can be used to illustrate both theological and pharmacological principles. In this particular instance, we're going to be looking at the mechanisms involved in neurotransmission, involving the sympathetic nervous system in the case of the today's demonstration. Now, we're also going to be looking at some pharmacological mechanisms. We will look at the effect of cocaine on responses to sympathetic nerve stimulation, and we will also look at the effects of cocaine on the responses to certain sympathomimetic amines. Now, before we get into the actual experimental procedure, I think it would be helpful if we took a look at the way in which we set up the experiment and the way we maintain it. We have a schematic representation of the experimental procedure. We anesthetize a cat with Nimbutal and then open a thoracic cavity and expose the right stellate ganglion, which is situated at the head of the first or second rib. Coursing from the stellate ganglion is the cardioaccelerator nerve, which travels to the peritracheal plexus, there joining the vagus nerve, and then innervates the atria and portions of the ventricles. We isolate the entire tissue mass, remove it from the thoracic cavity into a dish containing Krebs physiological solution at 37 degrees and aerated with oxygen and CO2. At this point, we clean up the atria, that is, we remove lung tissue, ventricle tissue, adhering fat and blood tissue, and clean up the ganglia and the nerve as well. From this dish, we then take the tissue and put it in a tissue bath, again maintained at 37 degrees, aerated with oxygen CO2. The contraction of the atria are monitored by a force displacement transducer, which converts the mechanical contraction energy into electrical energy, which then is coupled to a polygraph where we obtain a direct readout of the atrial force and rate of contraction. Now, in this portion of the demonstration, we are going to be concerned with the responses to nerve stimulation. And we are going to attempt to modify these responses by adding an agent which is cocaine. Now, before we actually obtain some responses, let's take a quick look at what we might expect in the way of responses to nerve stimulation from two separate atria. We have polygraph recordings. Stimulation is turned on at this point in this preparation. The initial rate of 91 is rapidly converted to an increased atrial contraction rate in the order of 162. However, it's interesting to note that there was no change in the actual force of contraction. Now, in contrast to this preparation, the lower recording, taken from another atrial preparation, shows that stimulation turned on at this point, the initial rate of 91, converted to a rate of 169 beats per minute, although in this case the contraction force was approximately doubled. Now, we can therefore expect, in response to sympathetic nerve stimulation, an increase in the rate of contraction and a variable change in the force of contraction depending upon the particular preparation we're dealing with. We can briefly take a look here at the apparatus that is involved in obtaining the responses. The central portion of the apparatus is the tissue chamber. That is, it's the bathing chamber in which we place the atria and maintain them. The atria are situated in Krebs bicarbonate solution, which is maintained at 37 degrees and vigorously aerated with a mixture of oxygen and carbon dioxide. The bottom atrium is tied to a stationary glass rod, and the top is tied to a string, which is then connected to the force displacement transducer. This transducer, in turn, connected to the polygraph, where we obtain a readout of the atrial force and rate of contraction. There appears to be arrhythmias, although often these convert into normal atrial contraction with time. Now, the rest of the apparatus consists of a chamber or of a reservoir to keep the Krebs bicarbonate solution. We keep this up high so that the fluid will, by gravity flow into the atrial bath. Before the fluid flows to the bath, however, it passes through coils which are immersed in a water bath maintained at 37 degrees. So that before the fluid reaches the atrial bath, it is fully equilibrated at this temperature. Now, we are able to add drugs directly into the atrial bath just by injecting them with a syringe and obtain any desired concentration of drug that we wish and obtain the response in this way. It is also possible to stimulate the nerve. In this case, in this portion of the demonstration, that is what we will be doing. We have an electrode, which is a double-pronged platinum electrode, situated at the surface of the bath, with the nerve lying over the two prongs. We apply stimulation to the nerve via this grass stimulator, and we are able to use stimulation of various frequencies, voltage, and impulse duration. Now, we can proceed with the experiment, and initially we will apply stimulation at a low stimulation frequency. The lower range of sympathetic discharge in most animals is on the order of 1/2 to 1 impulse per second. Therefore, we are going to look at a response to stimulation at 1 per second. In this and in subsequent stimulations, we are going to use 100 impulses, so that we will therefore apply stimulation at 1 per second for 100 seconds, so that we can now apply stimulation and obtain the responses. The stimulation is now on at 1 per second. We should not see a very rapid onset of the response at this low frequency. We will watch it only for 20 or 25 seconds, and then come back after the full time period has elapsed and look at the overall response. We can now take a look at the full response to stimulation at 1 per second for 100 seconds. As you can see, before turning the stimulation on, we had an initial baseline contraction of 111 beats per minute. Stimulation on at this point, then as we progress, the atria not increasing very much in rate, and after about 30 seconds, some arrhythmias, marked arrhythmias set in, although these convert back to a normal rhythm at about 50 seconds. Now, when we turn stimulation off at the 100-second time, the contraction rate was 123 beats per minute, therefore giving an overall increase of 12 beats per minute, about a 10% increase. After 10 seconds after turning stimulation off, the rate was around 115 beats per minute. Now that we've seen the response to 1 per second, we will take a look at the responses that we can obtain using a stimulation frequency of 5 per second. We are going to this time give stimulation for 100 stimuli, therefore giving us 20 seconds. Stimulation frequency is on 5 per second, and the stimulation is now on. This time we can expect to see a more rapid onset of the rate response, and in fact it is increasing. There's little change in the force of contraction. All right, the stimulation is now off, and we will see how rapidly it returns back to its baseline stimulation. We can see at this frequency arrhythmias are being maintained, although they are beginning to lapse back into a more normal rhythm slowly. Now, we have obtained a response at 5 per second, and then we went ahead and also looked at a response to a stimulation at 10 per second, and we can look at the results of all three of these stimulations and compare them, looking at the data we have on this chart here. We can see that when we stimulated at 1 per second, we had an overall increase in 12 beats per minute. The stimulation that you have just seen at 5 per second, again for 100 stimuli, gave us a final rate of 140 beats per minute, or an increase in 25 beats per minute. Now, the stimulation at 10 per second took us up to 206 beats per minute. Now, incidentally, this is near the maximum that the preparation is capable of contracting at, which is on the order of 220 to 225 beats per minute. Now, it's interesting to note the duration of the response after turning stimulation off. In this last column of figures, which are the rates 10 seconds after stopping stimulation, we see that at 1 per second, the rate came back towards normal fairly rapidly, and at 5 per second, we were back towards normal, 128, although not as close to it as we were in this case. However, at 10 per second, after 10 seconds of stimulation, the rate is still near the maximum that was obtained during this response, that is 200 beats per minute. Now we are going to proceed with the experiment and place cocaine in the bathing media. And we are going to put cocaine in the Krebs solution reservoir and expose the preparation to this drug so that from this point on, the preparation will be exposed to cocaine continuously. We will wait about 30 minutes and then obtain responses at these same stimulation frequencies again and compare the responses to those obtained before cocaine. We can then come back to the preparation in about 30 minutes. After the atria had been exposed to cocaine for approximately 30 minutes, we proceeded to obtain the responses to stimulation at 1, 5, and 10 per second again. We have the example of the response at 5 per second, and we can take a look at it and compare it with the same stimulation done before cocaine was put into the bath. The top recording is the control or the non-cocainized preparation, and the bottom record is the response obtained in the presence of cocaine. You remember in the control stimulation, the increase in the response was 25 beats per minute. In the presence of cocaine, however, the atrial contraction rate increased from 107 to 187 beats per minute. This is an overall increase of 80 beats per minute. And notice also that the increase in the contraction rate was maintained at its maximum response 10 and even 20 seconds after stopping stimulation at this point. The responses to 1, 5, and 10 per second are presented on the chart that we have here, and we can compare these results with the control preparation. After adding cocaine, 2 micrograms per milliliter, we see at 1 per second that the increase in the contraction rate upon stimulation, again for 100 seconds, was 51 beats per minute. This is about 4 times higher than the increase of 12 beats per minute at 1 per second previously. Now at 5 per second, as we have just seen, an increase in 80 beats per minute, and at 10 per second, an increase in 93 beats per minute. Again, we're getting up near the maximum response, although not yet to it. It's also interesting to note, again, the duration of the response. In each case, 1, 5, and 10 per second, the rates were maintained 10 and even 20 seconds after stopping stimulation. We have seen then that cocaine is able to potentiate the responses of the atria to nerve stimulation, and that it appears to, in addition to this, prolong the response after stopping nerve stimulation. In the second portion of the demonstration, we are going to be looking at some effects of sympathomimetic amines on the responses in the atria. We are also going to look at the effects of cocaine on these responses. Let's take a look at the drugs that we are going to be using. First of all, we will look at the responses of the atrial preparation to norepinephrine, the naturally occurring sympathetic transmitter that is located in the nerve terminals. We're going to compare this with the effects of isoproterenol, which differs from norepinephrine only in the fact that there is an isopropyl group attached to the nitrogen. Now we're also going to inject tyramine into the bath. This is a phenylethylamine derivative which acts to cause a response in the atrial tissue by displacing catecholamines, namely norepinephrine, from the sympathetic nerve terminal. Of importance in the difference in the actions of norepinephrine and isoproterenol is the fact that isoproterenol is not actively taken up into the nerve tissue as is norepinephrine, which in the case of norepinephrine is the primary mode of inactivation of the liberated transmitter substance. Now, we have prepared a fresh atrial preparation for this portion of the experiment. It no longer has the nerve tissue, and you're probably able to get a better view of the contraction of the atria. Now, for the first injection, we will give sufficient norepinephrine directly into the bath to obtain a concentration of 1 nanogram per milliliter within the tissue. We can inject the agent directly into the bath like this. The norepinephrine is now in. And we can observe if anything happens on the atrial contraction readout on the polygraph. Now with the sympathomimetic amines, usually some time is required before the full response sets in, usually on the order of 3 to 5 or 6 minutes. Of course, this depends on the dose. With a higher dose, much shorter time is required. I think we can come back to the full recording after 5 or 6 minutes and then look at the response. We have now obtained the full response to norepinephrine, the concentration of 1 nanogram per milliliter in the bath. The initial contraction rate was 125 beats per minute. Injection at this point, and then after approximately a minute, the rate was 136. Now, at readings after every 30 seconds, we reached a maximum of 143 at about 2 minutes. It maintained this for a minute or so, and then began to fall off back towards normal. Washing the bath at this point to remove norepinephrine from contact with the tissue, the rate then returned to normal at 125 beats per minute. We are now going to inject tyramine into the tissue bath, determine the response to this amine. Remember that this amine acts by displacing catecholamines from the nerve terminals. We're going to give tyramine so that the final concentration is 1 microgram per milliliter in the bath. The tyramine is now in. We've used a relatively higher concentration, and we should see the rate response, the onset of the rate response should be more rapid than that with norepinephrine. So you can see there's an increase in the force of contraction and the rate of contraction as well. The response that we obtain with this dose of tyramine will probably be near the maximum response of the tissue. In this particular preparation is on the order of 220 beats per minute. All right, I think we've seen the response to tyramine in this case, and what we will do now is obtain the maximum response to this drug. Then we will wash the tissue out, and after it comes back to a baseline or the control contraction rate, we can obtain a response to isoproterenol. We've obtained the response to tyramine, and we also went ahead and injected isoproterenol into the bath and obtained a response to this amine. We have the results of these 3 injections summarized on this chart. We can take a look at these and compare the responses. With norepinephrine, 1 nanogram per milliliter in the bath, there was an increase of 18 beats per minute. With isoproterenol, 0.4 nanograms per milliliter, the increase in the contraction rate was very similar, 22 beats per minute. However, with tyramine, at a concentration of 1 microgram per milliliter, we have a near maximum response, reaching a contraction rate of 214 from an initial of 117, to give us an increase in 97 beats per minute. The maximum response that we have observed in this preparation was actually 216 beats per minute, so that we have essentially a maximum response with tyramine. Now we are going to put cocaine in the Krebs reservoir so that the atria, again, as in the first part of the demonstration, is exposed to this agent for the remaining portion of the experiment. We will then obtain responses to these same sympathomimetic amines again in the same dosages that we used, and see how this agent, cocaine, modifies these responses. The preparation has been exposed to cocaine now for about 40 minutes. The cocaine is in a concentration of 1 microgram per milliliter. We're going to start the injections of the amines then by giving, again, tyramine at the same concentration as we did previously. We will again inject the tyramine directly into the tissue bath. The tyramine is in now. Now, if you remember from the last injection of tyramine, the onset of the response was quite rapid, and within 40 to 60 seconds we had a near maximum response. Well, it's been about a half a minute now, and the response is still very similar to the initial contraction rate. There appears to be very little response in this case. There may be a slight increase in rate and force beginning to set in. I think we can come back after we have allowed sufficient time to obtain a response if there is going to be one. We have now obtained the full response to tyramine, and then we proceeded and obtained the responses to both norepinephrine and to isoproterenol again. We have the results of these responses again on the chart, and we can compare the results obtained after cocaine at 2 micrograms per milliliter, not 1 microgram per mil as I stated previously. We can compare these responses with the responses obtained before adding cocaine. The initial baseline is somewhat lower after we added the cocaine, 106 to 107 compared with the 125 to 117 before. Injection of norepinephrine at the same concentration, 1 nanogram per milliliter, gave us an increase from 106 to 144, or 38 beats per minute. This is about twice the previous response of 18 beats per minute. With isoproterenol, there was actually very little change in the response, going from 107 to 125, giving a change of 18 beats per minute compared with an earlier change of 22 beats per minute. A great difference is seen in the response obtained with tyramine, going from 107 only up to 115, as you saw on the polygraph recording. The change here of 8 shows that the response earlier of 97 beats per minute was essentially abolished, at least greatly reduced, due to the presence of cocaine. How can the result of the demonstration that you have seen today be interpreted in terms of adrenergic mechanisms? I think the use of this diagram will enable us to view in a more sophisticated manner the actions of the amines and the actions of the drugs on the overall adrenergic system. In the diagram are presented the post-ganglionic sympathetic nerve terminal, indicated by this area, the synaptic cleft area into which the neurotransmitter substance is liberated, and here the effector organ or effector tissue. The small X structures represent the receptors themselves. The nerve terminal is surrounded here, indicated by this line, by the neuronal membrane. The norepinephrine, that is the sympathetic transmitter substance in the adrenergic system, is synthesized, stored in, and released from the sympathetic nerve terminal. The material is released into the synaptic cleft, where it then interacts with the receptors on the effector organ to bring about the physiological response. What is the fate of the noradrenaline, the NA, or norepinephrine, that is released into the synaptic area? There are several things that can happen to this norepinephrine. First of all, it can interact with the receptors. It can diffuse away from the synaptic area. This appears through a variety of experiments to be less important as a means of inactivation than our other mechanisms. Another mechanism for the inactivation of the released neurotransmitter substance is the enzymic degradation by the extracellular enzyme catechol-O-methyl transferase. However, of more importance in the inactivation of the released amine is the highly efficient reuptake process. This process causes the norepinephrine in the extracellular or synaptic space to be transported across the neuronal membrane by a transport pump. Once back inside the nerve terminal, the norepinephrine can be inactivated by the enzyme monoamine oxidase, which is located or associated with mitochondria, or it can be transported into these intracellular storage granules. Now, these storage granules are the site where the major portion of the intraneuronal norepinephrine is stored in a physiologically inactive form. It appears that it's stored as a complex between with ATP and with some macromolecular substance that is non-diffusible. Now, let us ask the question, how are the various physiological and pharmacological results that we obtained today, how can they be explained in terms of this overall structure? First of all, let us consider the responses to nerve stimulation. The passage of an impulse down the axon reaches the nerve terminal and causes the secretion or the release of the stored norepinephrine into the synaptic area, where it then interacts with receptors. We noticed several things about responses of nerve stimulation to low frequency and to high frequency stimulation, always using the same number of impulses. The response to high frequency nerve stimulation was greater than that to the low frequencies. Why is this? Even though we gave the same number of impulses. Well, it appears that as the norepinephrine is released from the nerve terminal during the low frequency stimulation, the reuptake process, the primary process for its inactivation, can keep up, or it can keep pace with the release process, so that during the stimulation period, a high concentration of amine is not achieved. Now, in contrast to this, during the high frequency stimulation, the catecholamine is released much faster, and it saturates the uptake process by reaching a relatively high concentration in this area in a short period of time. Once the uptake process is saturated, the excess amine then builds up to a higher concentration and is able to act on the receptors. Now, this same mechanism is reflected also in the duration of the response after cessation of stimulation. You notice that after stopping stimulation with 1 per second, the duration of the response was very short. The response, the elevated rate, came back towards normal very rapidly. However, in contrast to this, after cessation of stimulation at 10 per second, the duration of the response was actually prolonged. Why is this? Well, you can easily see that if the transport process is saturated, there's going to be excess amine which is not inactivated. Therefore, even after stopping stimulation, there will still be free catecholamine in the area of the receptors, and thus bring about a more intense and a more prolonged physiological response. How does cocaine interact with this system and bring about its effects on nerve stimulation? Cocaine has the ability to very efficiently block this transport process. It blocks the neuronal uptake process for norepinephrine. Thus, its interaction with nerve stimulation should be obvious. If you prevent the reuptake of the liberated catecholamine back across the neuronal membrane, block this process by cocaine, you will cause, first of all, a higher concentration of norepinephrine to be attained in this area, in the receptor area, and you will also prolong the response, since the reuptake process, again, is the main process by which this amine is removed from this area. Let's turn our attention now to the effects of the sympathomimetic amines that we used in the demonstration: norepinephrine, isoproterenol, and tyramine. First of all, how do these agents interact with the adrenergic system as we have it shown here? Norepinephrine and isoproterenol both act on the receptors, as does neurally released norepinephrine. The basic difference, though, is that isoproterenol is not taken up into the tissue. Isoproterenol does not have the affinity for the uptake process. Tyramine, on the other hand, acts indirectly. Tyramine is taken up into the tissue, displaces the catecholamine in the storage sites, which is then released and acts in a normal fashion. How will cocaine influence the effects of each of these three substances? The interaction between cocaine and exogenously added norepinephrine is essentially the same as that for endogenously released norepinephrine. That is, cocaine prevents its uptake, therefore building up a higher concentration in the receptors, leading to a more intense and a more prolonged response. With isoproterenol, however, which is not taken up into the tissues, we saw that cocaine essentially had no effect upon its response. This can be explained by the fact that since isoproterenol is not inactivated by uptake, and this is the main way in which cocaine acts in potentiating the effects of norepinephrine, cocaine will not influence the response to isoproterenol then. And this is in fact what we saw. Cocaine has the ability to block the uptake of tyramine as well as the uptake of norepinephrine. Therefore, the access of tyramine to the stored norepinephrine is blocked in the presence of cocaine, and tyramine is therefore no longer able to displace the endogenous amine, and as a result, no response was obtained. We have seen then that we were able to use this physiological preparation to demonstrate two aspects of pharmacology. First of all, what is the physiological basis for the pharmacological responses? That is, what are the mechanisms involved in neurotransmission? And we have used drugs to reveal these processes. And secondly, what is the basic mechanism of action of some of these drugs? We were able then to couple the physiological system with the use of the pharmacological approach to obtain an idea of these mechanisms.